Hello everyone, welcome back to another session of regeneration biology, and we will continue with where we left in the previous class. And today we will try to understand how different PCGs, that is position control genes, are contributing to the interaction with the neoblast, and they give rise to the lost tissue part or damaged tissue part. And we also understand how ectopic organs can be formed. That means additional organ. Ectopic organ means, say, usually you have got two eyes. If I make another two more eyes, that is ectopic. You have one nose. I make another two more nose, that is called ectopic. They are organs, but they are beyond the usual number and they are out of place. Okay. So progenitor targeting and ectopic organs. So we have seen so far that there will be a variation in the gradient of anterior-posterior or dorsoventral gradient will happen after amputation. Say for example, let us assume a planaria has got 10 segments. Okay, segment 1 to segment 10, let us assume. And first three segments, anterior most three segments have got one gene. And say posterior three segments have got another gene, let us assume. Now, if this planaria is cut into half, so 10 segment now became 5 segment per piece. Now, this anterior 3 segment is not in 3 segments now. It will restrict to the anterior most 1 segment. Means it is pushed anteriorly. Why? Because this posterior part of this anterior segment will start expressing more of the posterior genes, so that the existing piece now readjust. Say you had a house which is having 5 bedrooms and now you are moved to 1 bedroom house. Then your 5 bedrooms, different allocations what you had, you have to do everything in that single bedroom. This is what the animal will do first go. Same way, as you can see here, there is a distribution of anterior. In this picture you can see blue colored and then become white and then become yellow colored. Now after a segmentation that happens, what you have seen here in this existing piece, you are having blue color, white color, and there is no dedicated yellow color yet. But after some time, PCG pattern is re-established. You have one blue color, one light blue, one white, one light yellow and deep yellow, or light brown or deep brown. Same thing happened in the other fragment also. This is what repurposing or re-patterning or re-establishment of the gradient in that tiny fragment. That is very important for pursuing further with regeneration. So PCGs are expressed in the muscle. Left you have got PCG RNA probe pool, which is red in color. And as you can see here, because these are all different muscle specific gene, collagen probe, and SFRP2, it's basically indicating one of the position control gene expression. And in this B pattern, bottom also, this is the topmost portion what you are seeing, and bottom portion is basically the cartoon where the re-establishment of PCG expression has happened. Now important thing you need to know is that the migrating neoblast will come and interact with this individual PCGs located on the muscle and differentiate. Like here you can see, normal this should have been a given tissue and this should have been another given tissue. Now this has, in the anterior fragment it is continuing to do, but eventually you see the posterior fragment is giving. This is, both these tissues are formed as it happened in here. Same thing happened here also. Here technically only this piece, posterior fragment will give rise to only given set of tissue. Now the same posterior fragment start giving this anterior tissue also. So this is the logic behind this differentiation. Now another gene that is very specifically controlled, like we have seen, soon after injury, Wnt1 is expressed from every side, no matter whether it is anterior or posterior. We know that anterior region, the head regeneration, Wnt signaling is not needed. But then Wnt is expressed there. Who is controlling it? So we will be confused. If Wnt is expressed, why not Wnt signaling is not happening? So NOTAM is preferentially wound induced at the anterior facing wounds, and NOTAM is a classic Wnt inhibitor. And NOTAM's induction is not very well known, but it is restricting to the anterior most pole. Hence, even if Wnt is expressed, it doesn't matter. Wnt signaling is not going to kickstart. Head can form. Credit goes to NOTAM. And NOTAM inhibits Wnt signaling to promote head regeneration. How NOTAM is selectively activated at the anterior facing wounds remains unknown. Without PCG pattern, regeneration in follistatin, we remember follistatin is a responding molecule. Like who will come and tell the planaria, oh, there is a body part is lost? No one will come and tell planaria. It is done through induction of follistatin. If you knock down follistatin, say you are not keeping follistatin RNA do its job, or protein production from this RNA using RNAi, if an anterior most portion fragment is there, that will remain as it is. It is not going to replace the lost posterior part. Means planaria did not know, yes, I am injured. So follistatin is the one which helps. Without PCG pattern, regeneration in follistatin in myody RNA animals. Myody also you should know, we know the muscles are the place where the PCGs are expressed. If you knock down myody or NKX1, which is longitudinal or circular muscle markers, you will not be able to get any PCG expression, because PCGs are expressed, position control genes are expressed in the muscles, which will interact with the neoblast and they give rise to a tissue. The motivation for the neoblast to differentiate into a given tissue types come from the PCGs, and PCG expressed only in muscle. But if you get rid of the identity of the muscle, then you cannot have PCG. If you don't have PCG, no regeneration. So that is the logic. Regeneration fails to occur either with the follistatin RNAi or myody RNAi. This requires longitudinal muscle fibers, because myody is expressed in the longitudinal muscle fibers. Another example is a MEK inhibitor. Just like how important the follistatin is, the ERK MAP kinase signaling pathway is important for the persuasion of the regenerative response. Follistatin tells, yes, there is a damage, we need to regenerate. But that doesn't cause regeneration. To regeneration to kickstart, you need to have the ERK signaling to go on. So if you treat with an inhibitor, just like how follistatin RNAi prevented regeneration, MEK inhibition also prevent the regeneration. However, most important thing is removal of the inhibitor does not lead to regeneration, because you prevented that initial urge. Say you were hungry during the breakfast time and no one gave you breakfast. Now dinner time I am bringing breakfast to you and telling, please have this breakfast. By the time you may be hungry, but you are not in a mood to have breakfast. Or rather, you can't call it as a, maybe some people, their stomach is full of gas and if they eat something, they'll vomit. They should, because they are hungry so much, they should be drinking some lime juice or maybe having some orange juice. They cannot, if they have idli, they will have severe stomach pain or vomiting, whatnot, because stomach is full of acid. So you need to pacify the stomach. Same way, if you block follistatin or you block MEK pathway, regeneration doesn't happen, and then you withdraw the drug and telling, okay, now you regenerate, I withdraw the drug. It doesn't happen. But if you make a fresh injury, you cut again, and then don't give any follistatin RNAi, don't give any MEK inhibitor, then you will be able to get regeneration. So what does it tell you? It tells you that wound signaling or wounding is essential for it to kickstart the regenerative response. Coincidence of increased neoblast proliferation with positional information leads to amplification of regional progenitor classes, even if the target tissue of the progenitor remains uninjured, like this here. You can see here, this is the normal planaria. Here in this planaria below the eye region, there is a damage that is caused. And this damage tells the animal that, decapitation has happened. Actually, nothing has happened. There is a damage below the eye region. So immediately the planaria thinks of making a new eye or new head, because it is damaged well below. So the positioning of the injury, say if you do a tangential cut like across this plane, if you cut it, the lowermost cut is here. Hence, animal thinks above part is, whole part is missing, although this side is perfectly fine. But since it's a tangential cut and this is the lowermost portion, the animal thinks remaining portion is missing. Of course, the PCGs come into picture during regeneration, but that is how, as you can see in this picture, it immediately start amplifying the eye progenitors, mainly because it thinks the damage happened below the eye region, hence eye is damaged. Positional information and stem cell combine to bring about regeneration. PCGs can influence neoblast specification. PCG RNAi can impact regeneration, so the cellular output of neoblast is at some level dependent on PCGs. PCGs are pretty much everything when it comes to giving a meaning to the neoblast. Zeta neoblast produce progeny cells throughout the body that transit through transcriptionally distinct stages over 6+ days as they migrate and enter the epidermis. So what we should understand here is when there is any damage that is occurring to a given tissue, it has to come from the neoblast, and the gradient must be established in the form of neoblast. Without this, it is not going to work out. So single cell RNA sequencing data, it was determined that the dorsal and ventral zeta neoblast express different regional genes, and this time dependent expression of PCGs decide, or that gives the message to the incoming neoblast that what tissue type it has to give rise to. The dorsal regional identity of zeta neoblast required for dorsal PCG, that is nothing but BMP4. BMP4, you know, the dorsalization or the dorsal meaning or message to a back of planaria is given by the BMP4, and ventral is by its inhibitor, that is the noggin. Therefore, the PCG expression from muscle impacts the spatial identity of neoblast, influencing their fate. So this is the interaction between the PCGs, neoblast and the gradient. Positional information and neoblast specialization together drives new tissue production. Every tissue damage sets in an alert, leading to massive neoblast proliferation. Blastema production at the injury site and migration of neoblast happens in situ. Interaction of neoblast has to happen now with the PCGs. Morphogenesis ensues in a need-based pattern. What is the tissue that is damaged? What is the tissue that is missing? Accordingly, it will give rise to a new cell type or a tissue type. Research findings show that large wounds can amplify many progenitor types, even their target organ remains as it is. Like you saw in the previous picture, I can show once again. Here, there is a damage, big injury happened, but the target organ eye is as it is. Nothing has happened to the eye. So even when their target tissues remain, and suggest positional information combines with fate specification of stem cells to promote regeneration. So the identity of an injury or the nature of an injury is understood by the animal based on which place, which part it is damaged. Or in other words, if I got a wound in my chest, the organism thinks head is missing because chest is below the head region. Although head is there, it will immediately start to make the head. You can see here wound architecture, migratory cues and extrinsic, they are extrinsic factors that are influencing for the movement of the progenitors, especially how the cones or how the poles of the tips are forming, or the newly formed blastema is integrating with the existing anterior-posterior axis and dorsoventral axis, as you can see in this picture. And this will lead to a self-organization and produce a blastema pattern. Otherwise, what can happen? If you cut it in an angle, say you cut the planaria in this angle, you don't want a new head formed in this way. It look like the planaria look like bent like this. You want head like formed like this. You don't want planaria head to form like this. So unless it is aligned with the this plane, that's the central midline. So this cone, the anterior pole, is decided by this induction that is occurring here, and of course it is connected with the gene expression, and then there is an anterior posterior pattern that is established. So in top, what you can see, the anterior pole that promotes ML and AP, mediolateral and anterior posterior head blastema pattern. Bottom wound architecture cues determine the point of anterior pole formation connecting blastema pattern to the pattern of the pre-existing tissue. It has to gel well. It's just like making tunnel. You may have seen many people, they will make tunnel through the mountain. They start from one end and the other end. If they don't touch each other, they will end up making two parallel tunnels. But the alignment has to happen such a way that they will meet at a central point so that you save time. So this fusion is very important. So if your planning of the tunnel or the blueprint of the tunnel was perfect, it is going to meet each other. So the pole progenitors decide how and why it should be formed. Let us think about an example. This is NKX1 RNA. NKX is a marker of circular muscles. And if you knock down NKX, PCG expression get affected. And this NKX RNA, instead of having an anterior pole, you are having a spread out movement of the neoblast, or this cone is not defined. This will eventually lead to two head formation because there are two cones and both of them are trying to align with the midline and the lateral line. This DV median plane and the median line, both together is confused and you end up getting two heads. That is the issue that you can make with the NKX RNA. And self-organizing and extrinsic targeting cues govern the migratory behavior of the progenitor. So progenitors are waiting for the migration and they are just waiting for the proper PCG expression. But the PCG expression in turn depends on the muscles, and muscles quality depends, or the nature of their housekeeping genes such as MyoD or NKX1 decides whether a muscle remains as muscle. If a muscle has lost its identity, you can't expect a PCG expression happening. Hence, you can't expect a proper formation of a newly formed tissue. In this case, NKX RNA, it ended up getting two poles because circular muscles are contributing to the formation of the correct poles. And that got disturbed and you end up getting two heads. So progenitors' final establishment at the destination. How the progenitors resolve this dilemma of PCG discordance was demonstrated by simple experiment. PCG discordant means if PCGs are not properly expressed, there will be confusion that is occurring. When a head was amputated, the following a 3-day delay, that is time for allowing PCG resetting. One eye was removed. You take the animal and remove one eye. Remember, even if the entire anterior half is removed, the newly formed blastema will have the eye spot and the head that is formed, and later the head is moving forward. If you remove one of the eye, it just forms after 3 to 4 days, there will be an eye spot that will be formed, and you are removing one eye, you are plucking it. And when this eye regenerated, once you remove the eye, naturally eye will form. We have seen it. Even in adult, if you remove one eye, the eye will be formed right in this place. But here removing is slightly different. Why? The already formed eye is in a blastema. Blastema is dynamic. It is keep growing. In that you removed one eye. Then what will happen? The newly formed eye is going to be anteriorly shifted. What could be the reason? Because when the progenitors are coming, the existing eye will sequester. The other eye cannot sequester because eye is missing. It has to make the eye and then sequester. So then it is keep moving forward the minimum cells, and since the blastema is growing, the newly formed eye will be anteriorly shifted. So one eye will be here, another eye will be here. Normal eye is here, another eye is here. This is what happens. The progenitor targeted two different locations in the two sides of these head fragments. The remaining eye on one side, means untouched eye, acted as a self-organizing attractor that trapped incoming progenitors, making the progenitors not available for, like you may have seen it. If you are putting food, you have two chicken or goat or any animal in your house, you are putting them together, both will eat together. But you are not allowing one goat to eat. The other goat is not going to, or other chicken is not going to spare. It will eat up all the food. And when you release whatever leftover, only that chicken or goat will get it. So if this ate 1 kilo, the other will get only quarter kilo. Rest, this ate 1.75 kilo, the other goat. So this is what happened with the progenitors also. So the remaining eye, they trapped and they failed. The progenitor failed from reaching their TC, that is targeting zone. TC stands for targeting zone, where the actual differentiation must happen. Shifting the eye targeting zone medially and anteriorly by combining a parasagittal amputation, like you are doing in an angle cutting, to the side of one eye with decapitation allowed the progenitors to bypass the self-organizing influence of the remaining eye, resulting in the formation of a third eye. Say you cut it in an angle, existing eye is untouched, then the newly formed blastema assumes both are missing, you end up getting extra two eyes. So if you removed only one eye through one parasagittal section, the existing eye will remain as it is. Despite having one eye, since you made a plane below the eye region, then you will end up getting new blastema which will have two new eyes, and you will have the one old eye and two new eye. Totally you will have three eyes. Further, similar manipulations on the third three-eyed animal resulted in 4 and 5-eyed animals. So you can, while new eye is being formed, you can continue to cut. Already the animal has got 3 eye, you cut it in another angle, you will again end up getting another 2 more eye. Like you can make by plucking or by cutting in sagittal sections, parasagittal sections, you can end up making 3 and 4-eyed animals, 5-eyed animals, and extra eyes were stably maintained and targeted by the progenitor. This is something important. Animal doesn't lose a single eye, it will retain all of its eyes, producing stable alternative anatomies in wild type animals. Same way you can make extra pharynx also. Okay, so not that just eye, you can make other organs also extra. However, the beauty is that if you create a fresh amputation again you decapitated, got rid of all the 3 head or 5 head bearing eye, the newly formed head will have only 2 eyes. You will not have 5 eyes restored back. Future of anomalous eyes. Why are ectopic eyes maintained? Will they regenerate after damage to them? It was proposed that ectopic eyes were maintained simply because of their self-organizing properties combined with having access to eye progenitors. Eye are maintained in a specific region approximating the eye progenitor specification zone called targetable zone, and defined as a region the progenitors are capable of going for maintaining an organ. That is, in the newly formed blastema, 2 new eyes are formed. However, only eyes at the target zone are capable of being regenerated following resection. Spatially coerced progenitor specification is thus a central aspect of regeneration, allowing existing anatomy to have access to progenitors for their maintenance while positional coordinates are shifting. Similar experiments on PCG RNAi animals were a patterning phenotype resulting in 2 pharynxes allowed only 1 pharynx will be regenerated if you damage. If 2 pharynx are there, you cut one, new pharynx won't form because 1 pharynx is already there. Therefore, extra organs in incorrect location, that is ectopic organs such as eye and pharynges, can be maintained but not regenerated upon their removal. These observations indicate that self-organization and extrinsic targeting cues combine with a broad progenitor specification zone, that is TAS, to determine the destination of migrating regenerative progenitors. So there is a combination of multiple events, gradient, PCG, progenitors, everything has to come together for generation of a new organ. Like you can see in this picture, the amputations led to PCG shifting, but the self-organizing nature of the eye prevents the progenitors from reaching their target zone, whatever we discussed. That's what this is. This is one eye is been plugged, it got anteriorly shifted. And here you created a parasagittal section, and the newly formed blastema will always have the ability to, so you can continue to cut. Like if you cut it again, it will have extra eyes. This is the real-time picture of animals that have got 3 all of them have got 3 eyes extra, but you can create extra eyes if you wish, and enough medially to miss the eye that their migration, as you can see in this bottom, in this D panel, what you are seeing it. And E is what the wild type animals, as I already mentioned, having extra eyes, and F, this panel, that Wnt P2 and PTX7 RNA animals develop a second pharynx. The original anterior pharynx is maintained but not regenerated. So this is maintained, but if you cut it, the extra one ectopic organ do not come back, unless even for this animals, if it has got 3 eyes, if you cut it here or you get rid of the head, the newly formed head will have only 2 eyes. So a model for planarian regeneration, you can see here there are whatever we have studied is been described, that it has to involve the stem cells and stem cell fate specification, and the missing tissue response is essential. The target, the blind stem cell specification happens, and the positional information. I'm not going through each of this sentence because you can read it, because each steps are very important to understand, and they have to happen in a sequential manner. And then the positional information outside the progenitor is also important, because the muscles contain the positional information and the instructions must be stored in order to maintain, because the organs are revamped regularly. So, and would the signaling initiate the pattern of regeneration? Of course, the wound signaling contributes to the re-establishment of the pattern. Like we have seen, if you block the ERK signaling or you block follistatin, you will not get regenerative response. However, you can make them regenerate again if you make one more fresh cut. You make it, then it will continue to further regenerative angle. It will switch into the regeneration angle. Positional information impacts the fate of the stem cell depending upon where you are dealing with. Say you took, like we have seen it, if you block if you feed the animal with the Wnt1 RNA, you will end up getting ectopic heads that is being formed all around the animal. We have seen it. So that time what happens, the priority goes a absence of Wnt signaling. That is the priority that goes. It doesn't look which is the gradient you are talking about or which is the PCG you are talking about, because Wnt signaling is anti-head. Of absence of Wnt signaling, it becomes pro-head. So these logics you should put into picture, because it can also affect the gradient, anterior-posterior gradient. And organizers promote the blastema pattern. Organizers are very important. We have seen one of the classic organizers is the notum, which is giving the anterior-most positional identity. So progenitor migratory targeting, that is basically the specialized neoblast contribute for this progenitor's migratory targeting. And then self-organization of the progenitors, because sometimes what happens, these progenitors must organize itself. It encounters a differentiated target on its migratory path. It will immediately get into that organ. Like that is the cause, once you plug the eye from a blastema, the newly formed blastema is moving anteriorly shifted, because these progenitors are encountering an already existing eye. Hence, it will get migrated into that existing eye, because as a part of establishment of the existing organ or maintenance of the existing organ. So this enables the progenitor targeting to the remaining or partially injured body anatomy during regeneration. So these points should be kept in mind, that it's not just regeneration happens because of the congregation of all these factors. It also look what is there in the immediate neighborhood. If it finds an organ, it will get trapped. So this picture shows the model. That's the model for plenary regeneration involving positional information and stem cell. Like A, B, C, D, all this pattern, whatever we have seen is been discussed clearly. That means PCGs are important, and also the gradients are important, which in turn decides the PCG. And also how many hours after 0 to 6 hours what happened, 48 hours what happened, 70 to 96 hours what happened, 2 to 4 weeks what happens. That what are the changes that is occurring step by step manner, how anterior identity is maintained, how posterior identity is maintained, and how dorsoventral identity is maintained. Everything contributes to the formation of the organ. Like you can see here in this cartoon, this fragment and this fragment, it made a miniature head, this made a miniature tail within 70 to 96 hours. But it is not a full animal. It is making slowly and differentiate with the help of the gradient that is established in the animal. So we will study about another topic of regeneration in the next class. Thank you.